We had in mind the integration of different activities, upstream and downstream, even breaking a bit, the classical barrier between upstream and downstream. I would have probably talked to you about modeling, not yet machine learning and AI tools, because 20, 25 years ago, that was too early. And all those things at that time were not very well accepted.
All right, smart biotech scientists, buckle up, because you are in for a treat today. We are honored to have Professor Massimo Morbidelli with us. As a Professor Emeritus at ETH Zurich and Politecnico di Milano, and the co-founder of ChromaCon and DataHow, Massimo Morbidelli has been at the forefront of bioprocess innovation. He is currently the recipient of the ERC Advanced Grant Continuous Digitalisation of Bioprocesses at the Aristotle University of Thessaloniki in Greece. In this episode, we'll delve into the realms of integrated continuous biomanufacturing, cutting-edge chromatography techniques, and the future of recombinant protein manufacturing driven by machine learning. Get ready for an insightful conversation packed with groundbreaking insights and future-forward perspectives. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Massimo. It's a true honor to have you join us today.
Thank you, David. It's my big pleasure to be here with you today. And I look forward to some nice conversation.
Massimo, share something that you believe about bioprocess development that most people disagree with.
Well, you see, actually, if you would have asked me these questions about 20, 25 years ago when we started our activities in bioprocessing, I would have a lot of things to tell you. We had in mind, continuous processing versus batch. We had in mind the integration of different activities, upstream and downstream, even breaking a bit, the classical barrier between upstream and downstream. I would have probably talked to you about modeling, not yet machine learning and AI tools, because 20, 25 years ago, that was too early. And all those things at that time were not very well accepted. Many people were thinking these things are too complicated, continuous is very complicated, something goes wrong, how we can correct it. And then integration, why integrating, keep things separated, do things step by step. Something goes wrong, throw away everything and start again. Continuous is not so clear. So there was a lot of resistance. But today, all those concepts are much more accepted. We see a lot of continuous unit operations in the biopharma industry, even more than ever before. I was thinking to the bioreactors, for example. Perfusion had a brilliant career in the past years. Now there is a renaissance of perfusion bioreactor as well, not to talk about downstream, also a lot of continuous operation downstream. And we have the blooming, of course, of machine learning, mathematical modeling. Remember in the old days, this meaning 25 years ago, talking about modeling bioprocesses was thought to be not acceptable, right? It was accepted in the pharmaceutical industry of small molecules, but not for big ones. Big ones were too messy, too many components, impossible to describe. And now we have a lot of tools which can deal with this. So There is still a lot of way to go, but indeed there is a much more understanding and trust in this concept, and a lot of people is very high quality work. A lot of people is doing very high quality work to make progress, significant progress in this direction. And industry is pushing a lot. It's pushing a lot.
We're going to talk about all these exciting topics much more in depth today. So we're going to talk about the integrated continuous biomanufacturing, about chromatography, and also the industry trends such as machine learning and AI. But before we do that, Massimo, can you draw us into your long journey into the biotech world? Because you have a long career, and just highlight the key moments and decisions that shaped your career path.
Yeah, in fact, this brings me back 20, 25 years ago when we were very much into the chromatographic purification of small molecules, in particular chiral separation. Chiral separation has been a very challenging topic and a very good gym place for us chemical engineers and biotechnologists facing or entering in the pharmaceutical industry. So the simulated moving bed was the first continuous process based on chromatography for separating chiral component. And that was a very interesting concept. So chiral components are very similar, of course. So the selectivity that you can get with any possible stationary phase you can think of is going to be very small. 1.1, might 1.05, we're talking about this kind of numbers. So with such a small selectivity provided by the stationary phase, you have to have a very, very efficient process to achieve separation, maybe 98%, 97% with such small selectivity. In bioprocesses purification, we don't have that problem. We have a lot of molecules, very different molecules, and typically selectivity is very large. It's very, very hard to find situations where the selectivity is small. So the idea to use a more efficient process comes from the fact that we have now to improve the productivity, improve the yield, and in particular to be able to capture only the material, the molecule, the target that we want to purify, but all of it, don't lose it. And when you put these two things together, having high purity, but not losing, and high yield, right? So having one component, but only that one, don't catch any impurities, that becomes very difficult. And so therefore, we came with the necessity of a continuous process there also. And the idea then was, why don't we try to mutuate the concepts of simulated moving bed and develop the process for biomolecules? And that was beginning 2003, 4, 5. And the first guy who did the thesis on this was Lars Salman. I remember we sitting on the desk in my office and I was drawing the concepts that we should try to apply for having this applied to a three-component separation. The idea then was that with chiral separations, we have two components to separate. With our bioprocesses, we have three. So there is the central cut, which is the desired component, and then the impurity, some of which are heavier, some of which are lighter. So we have to have three exits. And in engineering, having two exits or three makes a lot of a difference. So we had to really come up with a new concept. And Lars was fantastic in really implementing all these ideas. And I remember walking in the lab and I saw him with 8 columns with all the pipes going all over the place. I hope you know what you're doing. I have no clue. But that was successful. And again, to show you how people did not trust really this concept at that time, we developed that process in the frame of a collaboration with a company. And the company said, well, interesting, too complicated for us. If you want to patent, you can do it. You give us a royalty, you give us a license to use it just in case, but we don't want to have nothing to do with it. And that was a big pharma, and that was the MCSGP, which is now known as a continuous chromatography for bio process. So that was the beginning. And with that, we started 2006, then came the patent. The company was started in 2005. In 2019, so 14 years later, the concept was so well understood and accepted, YMC decided to acquire our company, the ChromaCon, the spin-off of our activity. But at that point, or during that journey, it became clear that in order to have continuous processes to work, we had to improve our control strategies and automation concepts. And then machine learning came as the right tool to be used. And that was thanks to Alessandro Butte, who was a former student in the group in polymer reaction engineering, totally different concept. And he moved to Lonza, worked several years with Lonza, and then came back with this idea to implement machine learning ideas and concept to continuous chromatography and bioprocessing in general. So we did that, and he did that, and he had students like Michael Sokolov, first one, and then Fabian Feigl jumped into the game as well. And in 2017, we started DataHow, and DataHow now is only 7 years, but has already, I don't know, more than 40 people working for it, and has collaboration with 95% of the major pharma company in the world. And it is promoting this idea, which is digitalization. So digitalization means that we want to have now processes which are automatic and basically are able by themselves to reject disturbances and to find the optimal operating conditions. This is for the manufacturing side, and then a similar concept for the process development side. So that was really the journey that we took, and I think is going on still now. And I hope that we'll reach at some point our dream, which is to have very small production units, right? Fully integrated, continuous, and fully digitalized, meaning automatized. That is, in a nutshell.
Wow, what an amazing journey. Crazy also where you started and where you, quote, ended your career as a professor, because now you're officially retired, Massimo. But I'm curious, what project are you now working on? Like, can I say as a hobby, or is that still a profession, or how do we name that?
Well, David, in our case, it's difficult to separate the fun from the working. It's a job, but it's also a lot of fun. So in this case, I have to say that all what I told you before, as you know, was mainly about mammalian cells. So it was about products produced extracellularly, expressed extracellularly. That is also still now where everything is growing in particular. Now, after that, when I was close to retirement, actually, and so I retired in 2019 from ETH Zurich. I was 65, but in Politecnico Milano, I can go until 70. So I was still a professor in Politecnico di Milano. And then from there, I submitted this project to the European Research Council, which is about the continuous digitalization of bioprocesses. The big challenge here is now we do this with E. coli. So these are proteins which are expressed intracellularly, and we have in mind mRNA, oligonucleotides, and this particular project is on plasmid DNA. So this is the project which I submitted, and I got accepted the project a few months before retirement. These are so-called portable projects, so you can take the project where you want. And here I found the interesting collaboration with Professor Alexandros Kyparisides in the Thessaloniki University. So we decided to run this project. There is a 5 year project sponsored by the European Research Council. It's called an advanced grant. So it's about 2.5 million for 5 years. And so we have 4 students already running there, and we are putting together all the equipment. And here we have to face a lot of new problems, both with respect to continuous operation and digitalization. And in order to design and operate a unit for the continuous digitalized production of plasmid DNA, this is the goal of the project. And of course, I'm collaborating with that with the DataHow for the digital part. I'm collaborating with that with the ChromaCon or YMC ChromaCon for the MCGP, the continuous chromatographic purification. So all the old guys, are coming together in this project. It's a lot of fun. It's very nice.
Sounds like an amazing project. Now, for those who are not so familiar with continuous digitalization of bioprocesses, because the goal is to develop better and more robust processes. But what does this entail? Can you give us the simple version of what your project is about?
Simple version means the sequence of units, for example. So, yeah, the idea there is to have first a perfusion bioreactor. The cells will come out. The product is not the supernatant, like in mammalian cells, for example. And we take the cells, we have to go for the cell lysis in order to get the plasmid DNA out of the cell. And that, for example, is a unit operation which is currently not available commercially in a continuous form. So we are developing a continuous unit for cell lysis with Alexandros Kyparissidis. Then the product go further down. We need filtration units, and that we have some good ideas from different companies. And then the final purification, which is done by MCSGP, which comes from our experience with ChromaCon. So that is the flow of the units. The idea is to have a lot of PATs, right? Sensors all over the place that sends signals to the central SCADA system. And these signals are elaborated and transformed into measurements. And this go to a digital twin, which is the core of this digitalization effort, which will decide what has to be done to the plant in direction of, if there is a disturbance, how to reject the disturbances, or how to find a better, if not optimal, a better operating conditions for the plant. And the interesting part of the project that this digital twin is the same, which is developed at the process development stage of the entire production, right? So this will be able, for example, to use concepts like transfer learning or continuous learning, typical of machine learning, in order to optimize the manufacturing stage after having optimized the process development stage. But we may want to go more later on, maybe on this machine learning aspects of the project.
Yeah, absolutely. So just to summarize, so what you're doing is you're developing an end-to-end manufacturing process that is fully automated and fully running by itself using a digital twin, right? Correct.
Correct. And the digital twin is set up in the preliminary phase, which would be the one corresponding to the process design and development phase in a pharmaceutical industry.
So now this leads me to the next question. Using this technology, well, you have a lot of benefits, and one of them is that you will speed up process development and also reduce manufacturing costs. How does this new technology achieve these goals?
There are many ways of looking at this. So first of all is the continuous operation, which is automatized of each unit. And just because we are continuous and we are automatized, there are some advantages there. Second is the integration. So you can tell to the unit downstream what's happening to the unit upstream and optimize it in advance. So for example, you can say, okay, the titer is increasing to the chromatography unit, so watch out. In a few minutes, you will receive a higher titer, and the unit is ready to receive. And finally, the third point is the machine learning, the digital twin, which is able to tell to the unit, look, since the titer is increasing at the end out of the bioreactor, then the chromatography has to change the set points or the operating parameters in this and this way, so that when this happens, it knows what to do. So this is what happens, let me say, at the manufacturing stage. But before this, as you asked, in the process development stage, there is, for example, a technique which I really love and really fascinated, which is this transfer learning, which is a concept which for us humans is very obvious, right? So if you have developed 1, 2, 3, 4 bioprocesses for vaccines against a certain virus, and now a new variant comes about, what can you use of your experience to design the next process, meaning the process for the new vaccine for the new variants, by the minimum effort? And here the goal is to minimize the number of experiments that you need to perform in order to design your process, which means market time to market, which means amount of materials wasted and all advantages of any kind. So now, in our classical human life, this is done by giving the project to the guy who has done the previous processes and hoping that he can get good advantage of it. But sometimes the guy has left or something, he doesn't want to do it anymore or whatever. So these techniques help to use in the best possible way the information that has been gathered during the design of the processes for the previous vaccines to the design of the new vaccines. And here I have to say that I've been really surprised because everything here is based on machine learning, is based on statistics at the end of correlations. And so, for example, if you design to find the optimum of a process A and you have 20 experiments on this, so you take a model, feed the 20 experiments, and find the optimum. Now, if you take not only those 20 experiments of the same process, but you take 50 experiments of other processes, not process A, but B, C, maybe with a different cell line, different target, different protein, then you can reduce the number of experiments to find the optimum with respect to the previous case. You don't need all 20 experiments, maybe 10 are sufficient. Why? Because you are using a lot of knowledge which is inside those other experiments, even though they were not on the same process, they were on a different cell line, but still there is some correlation among the variables that are still remain valid in this context and then can be exploited. And this is what is done typically for language recognition or language translators or picture recognition. The difference is that those guys have a lot of experience. Pictures are very inexpensive, right? You can take as many pictures as you like. But one experiment for us in bioprocesses means maybe a fermentation run, which takes 2 weeks, and then you have all the analytical. So what we have to do is not to use a lot of experiments. Our goal is to use the tool of machine learning, of big data science, but not to take advantage of the big data, because we will never have big data in bioprocesses. We use the same concept of statistics, but to minimize the number of data which you need to achieve a certain goal. That is what is specific.
Hey, smart biotech scientists. Have you been enjoying our conversation with Massimo Morbidelli? We are continuing our conversation about continuous biomanufacturing, cutting-edge chromatography techniques, and the future of recombinant protein manufacturing driven by machine learning in Part 2. Part 2 will drop this Thursday, so don't miss it. Massimo Morbidelli is the Professor Emeritus at ETH Zurich and Politecnico di Milano, and he is a co-founder of ChromaCon and DataHow, and he's now currently the recipient of the ERC Advanced Grant Continuous Digitalisation of Bioprocesses in Thessaloniki. So what did you think about today's episode? Please tell us by leaving a review on Apple Podcast. I am looking forward to hearing from you and to see what you liked, what can we include the next time, what topics we should cover, and more importantly, we are able to reach even more smart biotech scientists like you. So see you on Thursday and have a wonderful week. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.